let's unpack this. Imagine a story that starts not really with a scientific hypothesis, but with an ancient legend, one shrouded in mystery, misfortune, the infamous Pharaoh's curse.
Right.
For centuries, you had these whispers of doom surrounding anyone who dared disturb the resting places of ancient Egyptian kings. People spoke of, supernatural forces, a vengeful past striking down intruders. But what if those whispers, instead of being purely supernatural, were actually, wow, a profound clue, a sort of breadcrumb trail leading us to one of the most surprising scientific breakthroughs of our time?
It's a fascinating starting point, isn't it? How myth can sometimes point towards a hidden reality.
Exactly. Today, we're taking a deep dive into the incredible, almost paradoxical story of Aspergillus flavus. Now, this is a common fungus, one that's kind of haunted humanity for millennia, from being the suspected culprit behind those historical curses to unbelievably emerging as a groundbreaking source of potential anti-cancer compounds.
It really is quite a twist.
Yes, you heard that right. From ancient threat to potentially modern cure, it's a journey that, well, it challenges pretty much everything we thought we knew about this microscopic menace.
And what's truly compelling here, I think, is how rigorous scientific inquiry has systematically replaced those anecdotal and mythological interpretations with verifiable mechanistic explanations.
Right, step by step.
Exactly. We're going to explore how this profound shift really highlights the sheer power of the scientific method. Its ability to demystify phenomena, advance our knowledge, and reveal nature's often unexpected and sometimes profound potential.
It's not just about a microbe, then.
No, not at all. It's about how our collective understanding evolves, moving from fear to understanding and ultimately maybe even to innovation.
So our mission today is to extract the most important nuggets of knowledge. We'll be looking at the latest cutting-edge research, some captivating historical accounts, and these surprising new discoveries that are completely reshaping our understanding of this notorious microbe.
There's a lot to unpack there.
Definitely. Get ready for some truly illuminating insights, maybe some aha moments, because this journey, from ancient Egyptian tombs all the way to modern oncology labs, well, it's nothing short of wild.
Okay.
So let's rewind our story. Let's go all the way back to 1922. Picture the scene. The thrilling moment when archaeologist Howard Carter, after years of, painstaking work, finally breaks through into Tutankhamun's tomb.
An iconic moment in archaeology.
Absolutely. And an archaeological triumph that just captivated the entire world. A discovery of unparalleled treasures, immense historical significance. But for many, especially the public eagerly reading the news, it also marked what they believed was the ominous beginning of the curse of the pharaoh's legend.
The media certainly played a huge role there.
Oh, absolutely. The press frenzy was immediate, intense, really driven by this thirst for sensationalism. It was the story everyone wanted to read. And the narrative of the curse, it truly exploded when Lord Carnarvon, who is the expedition's financial patron, tragically died in April 1923. That was just, 4 months after the tomb's opening?
Right, the timing was key for the narrative.
Exactly. His death was officially attributed to septicemia from an infected mosquito bite, which then progressed to pneumonia. But that one high-profile death, it sent shockwaves across the globe.
And the narrative of a supernatural curse just took hold immediately.
Instantly. It dominated newspaper headlines, captured the public imagination. It was just an irresistible story, wasn't it? Ancient powers striking back against those who disturbed their rest.
It plays into those deep-seated fears and fascinations.
Totally. And, it wasn't just Lord Carnarvon. The media's narrative was further reinforced by this string of other misfortunes, fatalities among the excavation team or their associates.
Right, there were several others mentioned.
Yeah, you had Carnarvon's nurse, for example, who supposedly died mysteriously, although details are often murky. His half-brothers, Aubrey and Mervyn Herbert, they also met unfortunate ends. Aubrey suffered from blindness, Mervyn from malaria-induced pneumonia.
So a range of different issues, really.
Yes, exactly. And then there was Hugh Evelyn White, who actually committed suicide and unsettlingly explicitly referenced a curse in his farewell note. That added a really chilling layer.
Wow, that's quite direct.
It is. And even Captain Richard Bethel, Carter's private secretary, was found dead later on. Now, whether these incidents were directly related or just tragic coincidences, well, they were all meticulously woven into the tapestry of the curse, by a sensation-hungry media.
Adding fuel to the fire with each incident.
Precisely. It layered the mystery. Now, while this modern curse was largely a media construct, especially for New Kingdom tombs like Tutankhamun's, it is worth noting that some truly ancient tomb inscriptions did contain explicit warnings.
So there's a historical basis for the idea of warnings, at least.
Exactly. They weren't always about, a supernatural plague or anything. More often, they threatened very real punishments like loss of honor, starvation, maybe lack of a successor. It reflected this profound desire to protect the sanctity and the eternity of the burial site.
Which makes sense from their perspective, protecting the afterlife passage.
Right. But all this, the historical warnings combined with the modern media frenzy, it really solidified the idea of a supernatural curse in the public imagination. A terrifying story passed down through generations.
And this compelling historical narrative, it raises a fundamental scientific question that really persisted for decades. Could there be a biological explanation? Something other than supernatural forces causing these seemingly inexplicable deaths?
Right. Could science offer an alternative?
Exactly. For a long time, the mystery remained pretty much shrouded in superstition. But then a pivotal event in 1973 provided a crucial opportunity for rigorous forensic investigation. This really began to unravel the true culprit.
Okay, what happened in 73?
This was the opening of the 15th century Casimir V Jagiellon tomb in Krakow, Poland. Now, what tragically unfolded there was eerily similar to the Tutankhamun narrative, but with a critical difference.
Oh.
10 out of the 12 researchers involved developed fetal lung infections and died within weeks of the tomb's disturbance.
10 out of 12, that's staggering.
It is. And while incredibly tragic, this incident provided a more, shall we say, controlled environment for scientific analysis compared to the circumstances in Egypt decades earlier. It allowed researchers to directly sample the tomb's atmosphere right then and there.
So they could look for biological agents immediately.
Precisely. And it was in this setting that Polish archaeologists, specifically a researcher named Bolesław Smyk from the Medical Academy in Krakow, isolated Aspergillus flavus in the tomb's air samples.
Ah, there it is, our fungus.
There it is. And media reports at the time immediately linked the fatalities to the potent aflatoxins produced by this fungus. This was a critical turning point. It shifted the focus from the mystical, the curse, to the microbial.
From superstition to spores.
Exactly. And retrospective analyses now strongly suggest that A. flavus may indeed have contributed to the premature deaths of some of Tutankhamun's tomb excavators, too. People like George J. Gould and Arthur Mace.
So looking back with modern knowledge.
Right. Though naturally, some doubt remains in particular cases. Lord Carnarvon's, for instance, given his specific medical diagnosis involving the mosquito bite and pneumonia. It's hard to be certain after so much time.
Sure, correlation isn't always causation.
Precisely. But the picture isn't limited to just A. flavus. Broader research has systematically identified a whole diverse array of other potentially harmful microorganisms and chemical hazards lurking in ancient tombs.
Like what else?
Well, things like Aspergillus niger, another common fungus. Various pore-forming bacteria like Pseudomonas and Staphylococcus have been found on tomb walls. And even insidious toxic gases, things like ammonia and hydrogen sulfide, which can build up in sealed environments.
Gases. Wow. What can they do?
They can induce severe respiratory and neurological effects, things like apnea, stopping breathing coma, even convulsions. So that further complicates the understanding of what might have afflicted those early tomb explorers. It might not have been just one thing.
A cocktail of potential hazards, really.
Potentially, yes. And we can't overlook another key biological factor: the documented presence of large bat colonies within Tutankhamun's tomb back in 1922.
Bats in the tomb.
Yes. And it's highly plausible that bat guano, which is a known reservoir for fungal spores, could have mixed with the pervasive desert dust when the tomb was disturbed.
Creating dust clouds full of spores.
Exactly. Creating a highly aerosolized environment, rich in fungal spores. This could have led to conditions like histoplasmosis, another fungal infection, and its symptoms: fever, enlarged glands, pneumonia. They closely align with Lord Carnarvon's clinical course.
Huh. So that's another plausible biological explanation for his specific case.
It is. It provides a compelling and scientifically sound alternative to the supernatural explanation for his demise. And if we connect all this to the bigger picture, it profoundly underscores how rigorous scientific inquiry systematically replaces those anecdotal or supernatural interpretations with verifiable mechanistic understanding.
It shows science chipping away at the mystery.
Absolutely. It exemplifies the sheer power of the scientific method in demystifying phenomena, turning those whispers of a curse into tangible biological explanations.
So, what does this all mean for us today? It seems, ironically, the very rumors and the sensationalism surrounding the Pharaoh's curse inadvertently catalyzed crucial scientific investigations into the microbiology of tombs.
It's a classic case of a problem demanding a solution. The intense focus forced a closer look.
Right. It created a problem that demanded a scientific solution, moving the narrative from just a spooky tale to a serious public health and archaeological concern. Archaeologists and historians now consider fungal spores not just a, fascinating biological curiosity, but both a significant preservation problem.
Because they damage artifacts, right?
Yeah.
Biodeterioration.
Exactly, causing biodeterioration of invaluable artifacts. And also a serious health hazard to conservators, the people who dedicate their lives to working on these ancient sites.
That understanding must have fundamentally changed safety protocols.
Completely. It's led to much more cautious and scientifically informed approaches for opening and preserving ancient sites. The British Mycological Society, for example, specifically notes that even modern events like that Casimir tomb tragedy are often linked to fungal spores, including those of Aspergillus flavus, a mold that produces aflatoxins.
So that knowledge is absolutely crucial now for anyone working in those kinds of confined ancient environments.
Definitely. It's standard procedure now to test the air quality.
And a remarkable aspect here is how this narrative shifted so dramatically. It moved from a purely mythological curse to essentially a cautionary tale for explorers and conservators alike. And that prompted deeper, more critical investigations into precisely how ancient burial environments can resurface dangerous pathogens.
So it's not just historical interest anymore.
No, it has real-world, tangible implications for how we interact with and preserve our past. It taught us that the real threats weren't, ancient spirits guarding tombs, but rather microscopic organisms that had been dormant for potentially millennia, patiently waiting for an opportunity, like the opening of a tomb, to become active again once disturbed.
That shift really exemplifies scientific progress in action.
It does.
This convergence of archaeology and mycology highlights a powerful lesson, doesn't it? Sometimes the most enduring myths, even those rooted in the supernatural, can inadvertently pave the way for the most significant scientific discoveries.
It's a great example of that.
It's a powerful reminder that curiosity, even when it's initially sparked by fear or superstition, can lead to profound insights into the natural world, unveiling its hidden complexities, its dangers, and ultimately, as we'll see, its potential.
Okay, so now let's zoom in. Let's take a much closer look at our main character in this dualistic story. Aspergillus flavus. What is this thing, really?
Right, beyond the tomb setting.
Exactly. This is an ascomycete fungus. Now, that's a large and very diverse group of fungi, and it has an almost worldwide distribution. You can find it pretty much anywhere on the planet where conditions are suitable.
Ubiquitous, then.
Very much so. It's typically saprotrophic. That means it thrives on dead plant material and, critically for us, on stored grains and other agricultural commodities, which is precisely why it's so common in agricultural settings and food storage facilities.
Okay, so that's its ecological niche mostly, decomposing plant matter.
Primarily, yes. Its species name, flavus, is Latin for yellow, and this refers directly to its characteristic yellow-green conidial spores, which are the reproductive units. They're readily visible in cultures or as a fuzzy growth on contaminated surfaces.
So if you've ever seen moldy corn or peanuts with that yellowish-green fuzz,
You've very likely witnessed Aspergillus flavus in action. Yeah. Microscopically, A. flavus is characterized by septate hyphae. Those are the thread-like structures forming the main body of the fungus, divided by cross walls and these globose conidiophores. Those are structures that produce and release its asexual spores called conidia.
okay. And reproduction, mostly asexual.
For a long time, the scientific community debated whether A. flavus even possessed a sexual stage, which is important for genetic diversity. But then, in a landmark study back in 2009, Horn and colleagues definitively demonstrated a sexual cycle in compatible strains.
Ah, so it can reproduce sexually.
Yes, leading to the designation of its sexual form as belonging to the genus Petromyces. Now, this discovery is hugely significant because it means the fungus can engage in outcrossing, essentially, exchanging genetic material with other individuals.
Which increases its genetic diversity.
Dramatically. It makes it much more adaptable to different environments and challenges,
Wow.
including, unfortunately, our attempts to control it. Ecologically, as we mentioned, it's best known for colonizing agricultural commodities.
Right, the grains and nuts.
Exactly. It's a pervasive and often silent threat. It infects crops both pre- and post-harvest. Think of staple crops like maize, peanuts, various cereals, tree nuts. It causes significant rot and widespread contamination that often goes completely unnoticed until harvest or during storage.
And you said it can be symptomless on plants?
Yes, interestingly, unlike some other Aspergillus species, A. flavus often remains symptomless on plants while they're still in the field. That makes its presence harder to detect initially. It also produces these asexual spore chains that are remarkably resilient to dryness, allowing it to persist in various environments, just surviving until conditions become favorable for growth again.
Very hardy.
Extremely. And within the species, there are also two major morphotypes, sort of like different forms. There are the L strains, characterized by large sclerotia. Those are hardened masses of fungal mycelium that act as survival structures, and they tend to be aggressively competitive.
Okay, the L strain.
And then you have the S strains. These have small sclerotia and are often much higher aflatoxin producers.
So the S strains are the bigger toxin concern.
Often, yes. Typically, L strains primarily produce B group aflatoxins, while some S strains are also capable of producing G group toxins, adding to their overall toxic potential. So there's variation within the species itself.
Okay, so this is clearly much more than just a historical curiosity found in ancient tombs. It's a globally ubiquitous fungus that actively infiltrates our food supply chains, often silently.
Hmm.
This widespread presence, coupled with its adaptability and resilience, makes its impact on public health and global agriculture a truly serious concern right now, extending far beyond those ancient curses.
Absolutely. It's a contemporary problem with massive economic and health implications.
And this leads us to where its true danger lies. Huge. In agriculture, its unchecked growth during crop drying and storage leads to the severe condition known as aflatoxicosis, if contaminated food is consumed by livestock or, more rarely but devastatingly, directly by humans.
And that can cause significant economic losses in livestock production, not to mention severe health issues for the animals themselves, liver damage, death.
Right. And clinically, for humans, A. flavus is recognized as an opportunistic pathogen. What does that mean exactly?
It means it primarily affects individuals who already have compromised immune systems or perhaps pre-existing conditions like lung disease. Healthy individuals can usually fight it off, but for vulnerable populations, it's a real threat.
Okay. And experts like Hedayati et al. Note that after Aspergillus fumigatus, it's the second leading cause of invasive aspergillosis. That sounds serious.
It is. That's a severe and often fatal lung infection. And A. flavus is also the most common cause of superficial infections among all the Aspergillus species, things affecting the skin, wounds, or the eye, specifically a condition called keratitis.
Eye infections.
Yes, fungal keratitis. It can be quite severe.
And its virulence, its ability to cause disease, is particularly significant, you mentioned.
Yes. Studies in animal models have shown that A. flavus spores can require a 100-fold lower inoculum, meaning a much smaller number of spores than A. fumigatus, to cause lethal disease.
Wow, so it packs a punch even in small doses.
It indicates its potent ability to infect and cause harm, even at low exposures. Common human syndromes caused by A. flavus include things like chronic sinusitis, which can be really debilitating, keratitis, that eye infection, which is especially prevalent in tropical regions and can lead to blindness if it's not treated promptly. And it also causes cutaneous or bone infections, usually following some kind of trauma where the fungus can enter the body through a wound.
And hospital outbreaks?
Yes, those are a concern too. They're often clonal, meaning they trace back to contamination from a single source within the hospital environment. That poses a major challenge for infection control in healthcare settings.
And beyond direct infection, there are allergies too.
Right. For people who are sensitized, exposure to A. flavus spores can trigger allergic reactions, induce severe asthma attacks, and other hypersensitivity responses. So it affects people in multiple ways.
Okay, so this extensive list of health impacts, it really begs the question, what precisely makes this fungus so dangerous? What's the mechanism?
The answer lies profoundly in its rich secondary metabolism, specifically its mycotoxins. These are among the most thoroughly studied fungal toxins known to science.
Secondary metabolism. So these aren't essential for its basic survival.
Not for its primary growth, no. They're complex chemical compounds produced by the fungus, often for its own survival and competitive advantage in its environment, maybe fighting off other microbes. But unfortunately for us, they happen to be highly toxic to humans and animals.
Okay. And the most infamous of these are?
The aflatoxins. They were famously discovered back in the 1960s. This happened during investigations of something called Turkey X disease.
Turkey X disease.
Yeah, it was a devastating and mysterious event involving the mass deaths of poultry, particularly turkeys. It was ultimately traced back to moldy peanut meal that had been extensively infected by aphids.
Ah, the contaminated feed.
Exactly. That incident truly put aflatoxins on the map as a global public health and food safety concern. Structurally, aflatoxins are a class of compounds called polyketide-derived diphenocoumarins.
Okay, that's a mouthful. What does it mean in simpler terms?
Well, without getting too deep into the chemistry, imagine them as complex chemical structures characterized by a distinct bisuranol system and something called a lactone ring.
Right.
Basically, these are intricate molecules with specific ring formations and chemical bonds that make them uniquely reactive and, unfortunately, profoundly potent toxins.
And they're classified as B and G types.
Right, that's a naming convention based on their blue and green fluorescence when you look at them under UV light during chromatography. Aflatoxin B1, AFB1 and G1 are particularly concerning because they have a double bond in a part of their structure called the furan moiety. This makes them highly reactive and therefore more dangerous.
And AFB1 is the worst.
AFB1, yes. It is notably the most toxic and potent hepatocarcinogenic liver cancer-causing natural compound ever characterized. Its hydrogenated forms, AFB2 and AFG2, are somewhat less toxic, but still definitely a concern.
How does it actually cause cancer? What's the mechanism?
It's quite insidious and actually well understood now. Once it's ingested, primarily through contaminated food, AFB1 is metabolized by enzymes in our liver, specifically cytochrome P450 enzymes, into a highly reactive intermediate called an 8-melamine epoxide.
Okay, an epoxide, that sounds reactive.
It is. This epoxide then binds covalently, meaning it forms a strong chemical bond directly to our DNA. It forms what are called AFB1 guanine adducts. It also binds to proteins. But the DNA damage is critical.
Why? What does the DNA damage do?
In humans and animals, chronic low-dose exposure to AFB1 leads to specific genetic mutations, particularly G to T transversions in codon 249 of the P53 tumor suppressor gene.
P53, isn't that a really important gene for preventing cancer?
Absolutely critical. It's often called the guardian of the genome. And this specific P53 mutation is frequently observed in individuals from high-risk regions for hepatocellular carcinoma, which is the most common type of liver cancer. This strongly links chronic aflatoxin exposure directly to the development of this devastating disease.
So the evidence is pretty clear then.
Overwhelmingly clear. Given this compelling evidence, the International Agency for Research on Cancer, IARC, unequivocally classifies aflatoxins as Group 1 human carcinogens.
Group 1, meaning definite causes of cancer in humans.
Correct. And epidemiological studies unequivocally link dietary aflatoxin to liver cancer, especially in regions where hepatitis B is endemic. The two factors together create a synergistic and even more dangerous risk profile for liver cancer development.
Wow, so chronic exposure is linked to cancer. What about high doses?
Acutely, yes. Very high doses of aflatoxin can cause a sudden and severe illness known as aflatoxicosis. This can lead to rapid onset of severe illness, even death. In animals, it can result in severe liver hemorrhage, biliary hyperplasia, that's an overgrowth of bile ducts, and rapid death.
And in humans?
In humans, though fortunately rarely reported today due to much better food safety measures in many parts of the world, acute aflatoxicosis can lead to severe jaundice, acute liver failure, and death. A truly devastating condition.
Okay. But aflatoxins aren't the only toxins this fungus makes.
No, as potent as they are, they're not alone. Other metabolites also contribute significantly to its overall toxic profile. There's cyclopentonic acid, or CPA. That's another mycotoxin that acts by interfering with crucial cellular pumps called calcium ATPases.
Calcium pumps.
Yeah, imagine these pumps as tiny, highly regulated vacuum cleaners meticulously managing calcium levels within our cells. When CPA disrupts them, it can cause a cascade of problems, manifesting as severe neuromuscular symptoms in affected animals.
Okay. Any others?
There's also steridomycin. It's itself a hepatocarcinogen, though it's usually present in lower amounts than aflatoxin. And another one, mitropropionic acid, inhibits mitochondrial enzymes, which are crucial for cellular energy production, and that can potentially cause neurotoxicity.
So it has a whole arsenal of toxins.
It really does.
Yeah.
And due to their profound impact on livestock and human health, many of these compounds are strictly regulated in agriculture worldwide. Regulatory limits exist for food and feed pretty much everywhere.
But contamination still happens.
Despite these comprehensive regulations, yes, aflatoxin and CPA contamination remains a serious and pervasive problem, particularly in developing countries. There, food storage, drying practices, and monitoring systems might be less robust, leading to ongoing public health crises.
Right. Infrastructure makes a huge difference.
Absolutely. Interestingly, in natural environments, A. flavus often coexists with a complex array of competitive microbes, and these other microbes can actively modulate aflatoxin levels. Intense fungal competition actually tends to suppress aflatoxin production.
Oh, really? So competition keeps it in check sometime.
It seems so. Whereas monocultures on host plants, say a field of corn where A. flavis dominates without that natural competition, can allow prolific toxin production. It highlights the complex ecological interactions at play.
So this humble, or maybe not so humble, yellow mold is far more than just a historical curiosity. It is a serious global threat, one that infiltrates our food systems, poses very real and current health risks to millions. It's truly a villain in our narrative so far.
A significant adversary, yes.
But hold on, because the plot twist is coming. This organism, which we've just established as a major adversary, is about to reveal a surprising and potentially heroic side. Okay, given everything we've just discussed, A. flavus as a notorious pathogen, a potent toxin producer, the very idea of it being a source of medicine might seem, well, completely counterintuitive, maybe even wildly improbable.
It definitely goes against the grain of its reputation.
Right. It's like discovering that a venomous snake holds the key to a cure for a debilitating disease. Yet history has repeatedly taught us to look for hidden potential in the most unexpected places. Fungi, after all, gave us one of the absolute cornerstones of modern medicine, penicillin.
From the common mold penicillium, a game changer.
Exactly, and that wasn't an isolated incident. Countless other life-saving drugs, from immunosuppressants like cyclosporine to cholesterol-lowering statins, have emerged from microbial sources, often fungi.
Nature's pharmacy is vast and often surprising.
It is, and this past success fuels an enduring optimism, doesn't it, about nature's untapped pharmacy.
Absolutely. And what's truly fascinating here is that despite this proven pharmaceutical capacity, scientists estimate that only about 5% of fungal biodiversity has been successfully cultured in laboratories.
Only 5%. Wow.
Think about that for a moment. 95% of fungal species are still largely unknown to us, let alone explored for their biochemical potential. It's staggering.
That statistic points to an immense, largely untapped reservoir of novel bioactive compounds, doesn't it? Stuff that could address critical unmet medical needs.
Exactly, including potentially entirely new classes of anti-cancer agents, or maybe innovative solutions for the growing crisis of antimicrobial resistance. Imagine the vast chemical diversity we're still missing out on, just waiting to be discovered.
That's a mind-boggling thought, the sheer scale of unexplored potential. So it sounds like the scientific community is actively seeking new frontiers. But how did this radical reevaluation, this turning point for Aspergillus flavus, specifically moving from foe to potential pharmacopoeia, how did that even begin? What were the first glimmers of hope?
Well, the journey to reevaluate A. flavus began with several recent studies that explicitly started looking for anti-cancer agents within its extracts. They were actively challenging the prevailing view of the fungus solely as a harmful entity.
So purposefully looking for the good stuff.
Exactly. For example, a significant study by Sager and colleagues in 2024 took an intriguing approach. They isolated a strain of A. flavus from a highly unusual environment, a Saudi Arabian saline marsh, a salty environment which might select for unique adaptations.
Okay, interesting source.
Very. They then tested its crude extract, essentially a raw mixture of all the compounds produced by this specific fungal strain, against multiple human cancer cell lines in laboratory dishes.
And what did they find?
What they found was quite striking. Significant cytotoxicity. That means a potent cell-killing effect. They measured an ICUR value less than 50 micrograms per milliliter against leukemia and other tumor cells.
Okay, explain ICUR again. Low is good, right?
Right. ICURs tells you the concentration of a drug needed to inhibit 50% of a biological process in this case, to kill 50% of the cancer cells. A low ICURs indicates high potency, so less than 50 BML was definitely a promising initial signal.
Okay, so the raw extract killed cancer cells. Did they know what in the extract was doing it?
That was the catch. Metabolomic analysis of this crude extract identified numerous known Aspergillus metabolites. Things like aflavanines, which are a type of indole alkaloid, formalagens, hydroxycygonic acid, chrysogene, it's a benzodiazepine-like alkaloid, and aspergillic acid, which actually has antibiotic properties.
So a complex
A
mix.
very complex mix. And critically, and somewhat problematically for thinking about drug development, the analysis also detected aflatoxins B1 and G1 within this crude extract.
Oh, the carcinogens were in there too.
Exactly. Sager and his team themselves noted that these aflatoxins, at the high doses present in the unpurified extract, likely contributed significantly to his overall lethality against the cancer cells.
So the good effect might have been partly due to the bad stuff.
Precisely. It meant the extract was a mixed bag, potentially beneficial compounds alongside definitely harmful ones. So while this work clearly implied that A. flavus extracts could kill cancer cells, it didn't isolate which specific molecule or molecules were responsible for the primary desired anti-cancer effect. It left the precise active actors unknown.
A critical hurdle for developing a targeted therapeutic.
Absolutely. It was a broad-spectrum hit, but it lacked the precision needed for a safe and effective drug. You can't give patients a crude extract containing aflatoxins.
Right, of course not. So it was a strong hint, a tantalizing clue that there was something more here, a hidden pharmacopoeia within this notorious fungus. Researchers had found a potentially valuable effect in this haystack of compounds, but they still needed to figure out exactly what the needle was and how to separate it from the toxic stuff.
A classic challenge in natural product discovery.
And then came the game changer, something truly remarkable that emerged from a 2025 study. This is where the story takes a really unexpected and exciting turn.
This was the groundbreaking discovery of fungal IPPs.
RPPs. Okay, break that down.
RPPs stands for ribosomally synthesized and post-translationally modified peptides. They were discovered in A. flavus by Nye and colleagues in 2025. Now, RIPPs are a rapidly emerging class of natural products. Think of them as tiny, highly complex protein fragments.
Okay, peptides, like small proteins.
Exactly. But they undergo special finishing touches or modifications after they're initially made by the cell's ribosomes. These modifications give them unique structures and often very powerful biological activities.
So they're not just simple peptides.
Not at all. Now, while thousands of RPPs have been identified and studied extensively in bacteria, their discovery in fungi has, historically, been remarkably challenging. Only a handful had been identified until recently.
Why so few in fungi compared to bacteria?
It seems it was largely due to previous misclassification and just a limited understanding of their unique biosynthetic pathways. They were essentially invisible, or very difficult to spot for researchers using traditional methods. The genes didn't look like typical peptide synthesis genes.
So new techniques were needed.
Exactly. The researchers at the University of Pennsylvania School of Engineering and Applied Science, led by senior author Sherry Gao and first author Keone Ni, employed a really sophisticated and innovative approach. They combined advanced genome mining.
Sifting through the DNA sequence.
Right, sifting through the fungus's genetic code looking for specific patterns. And they combined that with comparative metabolomics, which involves analyzing all the small molecules produced by the fungus under different conditions.
Okay, genetics plus chemistry.
Precisely. Through this meticulous process, they identified a unique gene cluster within A. flavus. And this cluster encoded something unexpected, a heptacyclic benzopyranoindolene peptide scaffold.
Whoa! Heptacyclic. 7 rings.
7 rings. They termed these resulting compounds asparagomycins, marking a significant new chapter in fungal natural product discovery. What truly sets these molecules apart is their unique and previously undescribed structural motif. It's characterized by what the researchers called interlocking rings.
Interlocking rings. Sounds complex.
Extremely complex. Specifically, they possess an intricate 7-ring architecture that includes a fused indole benzofuran's core. This extraordinary structural complexity isn't just an academic detail. It's believed to be fundamental to their potent biological activity and their distinct mechanism of action.
So the structure is key to how it works.
Very likely, yes. Now here's where the real ingenuity comes in. While the naturally occurring asparagomycins C and D showed some inherent activity and contained something called an N-terminal pyroglutamate, another form, asparagomycin B, was initially inactive in its native form. It didn't do much.
Okay, so the natural version wasn't the magic bullet.
Not asparagomycin B, no, But the research team had a brilliant key insight, a moment of true innovation. They decided to synthetically modify asparagomycin B. Specifically, they started appending various lipid chains, fatty acid chains, to its N-terminus, the beginning of the peptide.
Adding fat molecules to it.
It
Why?
was a hypothesis that this might help it get into cells or interact differently, and this seemingly small chemical modification dramatically enhanced its bioactivity and, crucially, its membrane permeability. It effectively transformed a dormant molecule into something remarkably powerful.
So the modification unlocked its potential.
Exactly. And the results were striking. Notably, one of these modified analogs, specifically compound 2L6, which had an 11-carbon fatty acid chain attached, exhibited nanomolar cytotoxicity against leukemia cells, specifically OCI-AML3 cells.
Nanomolar. That sounds incredibly potent.
It is. Extremely low concentrations were effective. And this potency was comparable to standard FDA-approved leukemia drugs like cytarabine or daunorubicin drugs that have been mainstays of leukemia treatment for decades.
Comparable to existing chemo drugs. That's huge.
It is. This isn't just a modest improvement. It's showing efficacy in parallel with established powerful drugs, but potentially offering a new chemical scaffold, which might help overcome resistance issues.
Wow.
This truly was a remarkable transformation, taking a dormant fungal peptide and turning it into a potent anti-cancer agent. It perfectly reflects the cutting edge of modern natural product chemistry approaches.
It really highlights the importance of not giving up on compounds, right?
Yeah.
And using new techniques.
Absolutely. It profoundly highlights the critical importance of reevaluating past assumptions about natural compounds and, crucially, developing new analytical and synthetic techniques to uncover previously overlooked classes of natural products. This opens up a vast, previously inaccessible chemical space for drug discovery,
Showing us that nature still holds countless secrets.
waiting to be unlocked with the right tools and, frankly, the right ingenuity.
So, from a previously forgotten corner of a common, even feared fungus, a powerful new potential weapon against cancer has emerged. What an incredible shift in perception. From ancient curse to modern potential cure. It truly shows the unexpected turns scientific discovery can take.
Okay, so now that we know asparagomycins are potent, especially that modified version, the next crucial question is, how exactly do they work at the cellular level? What is their precise target within the incredibly complex machinery of a cancer cell,
Right.
particularly given that intricate 7-ring structure?
What's them kill leukemia cells specifically?
Mechanistic studies, digging into how they work, showed that these compounds act by specifically disrupting microtubule formation.
Microtubules, aren't those like the cell's internal skeleton?
Exactly. They're essential cellular components. Think of them as the dynamic scaffolding or railroad tracks required for proper chromosome segregation during cell division, mitosis. They form the spindle apparatus that meticulously pulls duplicated chromosomes apart into 2 new daughter cells.
So, absolutely critical for cell division.
Absolutely. Without correctly formed microtubules, cell division simply cannot proceed correctly. And interestingly, many existing successful chemotherapeutic agents, drugs like the taxanes, such as paclitaxel, and the vinca alkaloids, like vincristine, they also target these very same microtubules to inhibit cancer growth.
Ah, so it's a known validated cancer target.
It is, but potentially hit in a new way by this new scaffold. Carefully designed cancer cell assays, those are experiments conducted in the lab using cancer cell lines, clearly demonstrated what's called a mitotic arrest phenotype in the treated cells.
Mitotic arrest, meaning they get stuck in the division process.
Precisely. Leukemia cells treated with asparagomycins formed abnormal spindles, essentially. Their internal scaffolding was faulty, and they couldn't complete cytokinesis, which is the final stage where the cell physically pinches off into 2 new cells. This effectively arrests their uncontrolled proliferation.
Stops them multiplying.
Exactly. As Sherry Gao, one of the senior authors, explained, asparagomycins are essentially putting a stop to the runaway, uncontrolled cell division that is the very hallmark of malignancy. They are preventing cancer cells from replicating themselves.
That's an incredibly precise mechanism then, jamming the engine of runaway cell growth. Instead of just broadly destroying cells, they're interfering with a fundamental process cancer cells rely on.
That's the idea, yes. Targeting a key vulnerability.
That sounds incredibly precise. But thinking about these complex cyclic peptides, you mentioned the lipid chain helped, but a common challenge for molecules like this is actually getting into the cells where they can do their work, right? Many large or complex molecules struggle with getting across the cell membrane.
That's an astute observation, and it's a critical point in drug development. How does the drug get to its target? And this leads us to another truly fascinating discovery within this study.
Okay.
They used a genome-wide CRISPR screen. Now, this is a cutting-edge genetic tool that allows scientists to systematically turn off thousands of genes, one by one, in cells and see what effect it has.
Like switching off lights one by one to see which one controls the fan.
Kind of like that, yes. And this screen identified a specific gene, the lysosomal transporter gene, SLC46A3,
Yeah.
as absolutely critical for the lipidated asparagomycin's entry into the cells.
SLC46A3,
Mm.
a transporter gene. What does that usually do?
Ki Ni, the first author, vividly described SLC46A3 as acting like a gateway. It facilitates their penetration into the cell's main fluid, the cytosol. This was profoundly significant because it explains a key, previously unknown step in the drug's journey. Normally, SLC46A3 functions as a transporter protein that primarily helps materials exit lysosomes.
Lysosomes, the cell's recycling centers.
Exactly. They collect, break down, and process foreign substances or cellular waste. But in this unique case, it seems SLC46A3 facilitated the transfer of the cyclic asparagomycin peptide from the lysosomes into the cytosol, where it can then exert its effects on the microtubules.
So it's using an exit door as an entry door into the main part of the cell.
That appears to be the case, yes. And the researchers provided compelling evidence. They found that knocking out, or deactivating, the SLC46A3 gene in leukemia cells rendered them completely resistant to 2L6, that potent analog.
So without the gateway, the drug couldn't get in to work.
Precisely. It definitively confirmed SLC46A3's crucial role in the drug's uptake. Now, this discovery is particularly significant for future drug development. It suggests that SLC46A3 might also facilitate the entry of other cyclic peptides with medicinal properties.
Opening up possibilities for other drugs too?
Potentially, yes. And this specific requirement for SLC46A3 might help explain the leukemia specificity we see with asparagomycins. Maybe leukemia cells express more of this transporter or rely on it more. And it suggests powerful strategies like that lipid conjugation to optimize drug delivery by perhaps exploiting other lysosomal transporters too. It opens up entirely new avenues for designing and delivering drugs that were previously thought undeliverable because they couldn't get into cells effectively.
So it's not just about finding the right drug molecule itself, but also finding the right key, like this SLC46A3 transporter, to unlock the cell's door that makes the difference. That's a huge insight, a real aha moment. It highlights the incredible ingenuity of nature and our growing ability to understand and leverage these intricate cellular mechanisms for therapy.
It's a beautiful example of basic science revealing therapeutic pathways.
Now, one of the biggest challenges, as we know, with traditional chemotherapy is the devastating widespread side effects. That's often because they lack precision, right? They harm healthy, rapidly dividing cells like hair follicles or gut lining alongside the cancer cells.
Yes, the off-target toxicity is a major limitation.
Do asparagomycins offer a better profile? Did they show more specificity? That would be a massive game changer for patients.
Indeed. Specificity is absolutely critical for any future medication, especially in oncology. And asparagomycins demonstrated this characteristic remarkably well, at least in these initial in vitro studies.
How so?
Notably, the compounds had little to no effect on other major types of cancer cells. They tested specifically breast, liver, or lung cancer cells, even at high concentrations.
So very targeted towards leukemia cells in their tests.
It appears so. And what's more, they also showed no activity against a wide range of common bacteria and fungi, indicating a very narrow and precise targeting, seemingly focused on the leukemia cells that rely on that SLC46A3 transporter.
That high degree of specificity, that's a crucial advantage.
Absolutely. It implies a potential for significantly reduced systemic toxicity, and therefore dramatically improved patient outcomes compared to conventional broad-spectrum chemotherapies. This could truly revolutionize leukemia care if it holds up.
Minimizing those harsh side effects.
Exactly. Minimizing adverse effects that often diminish a patient's quality of life and sometimes even limit their ability to continue treatment. It enhances the overall therapeutic index, that margin between the effective dose and the toxic dose.
And you mentioned it's a new scaffold.
Yes. Compared to conventional chemotherapy, asparagomycins offer a completely new chemical structure. While mechanistically they're similar to other tubulin-targeting agents like taxanes and vinca alkaloids, their novel structure means they might be able to bypass common resistance mechanisms that cancer cells often develop against existing drugs.
Offering hope for patients whose cancer has become resistant.
That's a key potential benefit, yes. And it's also incredibly important to note, circling back to safety, that the original fungal pathway for these peptides, the asparagomycins, likely does not produce aflatoxin.
Ah, right. Good point.
Asparagomycin B lacks the characteristic chemical structures, those bifuran motifs strictly associated with aflatoxins. So these peptides are chemically distinct from the dangerous aflatoxin biosynthesis pathway. It's absolutely crucial for their safety profile moving forward.
That's fantastic news. A targeted approach, potentially bypassing resistance, and seemingly separate from the toxin production. This could truly mean a much more tolerable and effective treatment for patients. Improving quality of life during treatment is huge.
That level of specificity is certainly what researchers and clinicians strive for.
So what's next for these incredibly promising compounds? And what are the inevitable hurdles that still need to be cleared before they could possibly become a reality for patients in a clinical setting?
Okay, the in vitro results for asparagomycins against leukemia cells sound incredibly promising. I mean, potency comparable to established drugs, high specificity. But translating that success, taking it from a petri dish into a real-world medication for human patients, that's always a monumental task, isn't it?
It absolutely is. It's a complex, lengthy, and unfortunately often challenging journey from the laboratory bench to the hospital bedside.
So what are the next critical steps in their development? What kind of journey awaits them?
Well, while the initial findings are strong and certainly warrant the excitement, the immediate next step involves rigorous in vivo testing.
Meaning studies in living organisms. Exactly. Typically animal models first. This is a non-negotiable phase in drug development, with the ultimate goal, of course, being progression to human clinical trials. These preclinical animal studies are absolutely essential to comprehensively evaluate several key aspects of the asparagomycins.
Like what specifically?
We need to understand their pharmacokinetics. That's how the drug moves through the body, how it's absorbed, distributed, metabolized, and eventually eliminated. We also need to determine its bioavailability, how much of the drug actually reaches its target site in the body in an active form. And crucially, we need to assess its full safety profile in a living, complex biological system, all before it ever gets near a human patient.
Testing for unexpected side effects in a whole organism.
Precisely. And it's crucial here to acknowledge the significant challenges inherent in translating that in vitro success in test tubes or petri dishes to in vivo and ultimately clinical reality. History tells us that a significant percentage of drugs that appear safe and effective in animal models ultimately fail in human trials.
Why is that gap so large sometimes?
It often happens due to unforeseen toxicity in humans, or perhaps a surprising lack of efficacy that just wasn't predicted by the animal studies. This discrepancy is frequently attributed to species-specific physiological differences. Animal models, while invaluable, simply may not accurately predict the precise human response.
Can you give an example?
Sure. Some medications considered remarkably safe in humans, like aspirin, might actually not pass animal testing if they were developed today due to specific sensitivities in common lab animals. Conversely, and tragically, other compounds that appeared safe in animal models have proven harmful, even lethal, in early human trials.
So, animal studies are necessary but not perfectly predictive?
Exactly. They are an absolutely necessary step, but their results must always be interpreted with a healthy dose of scientific caution. These studies are designed to reveal the full, complex picture of a drug's behavior in a living system, anticipating challenges that a petri dish simply cannot replicate. But they aren't a crystal ball for human outcomes.
So, the journey from petri dish to patient is indeed long, often fraught with challenges, requiring cautious interpretation and incredible scientific rigor at every single step. It's clearly not a straight line from discovery to cure.
Not at all. And beyond just the animal models, there are several other significant challenges that span various stages in the development of any fungal-derived anti-cancer agent, not just asparagomycins.
Okay, like what kind of broader challenges?
Firstly, we often face production and supply limitations. Remember that 5% figure. Only a tiny fraction of fungal biodiversity has even been successfully cultured in labs. That severely limits the initial discovery phase right off the bat.
We might be missing countless potential drugs just because we can't grow the fungi.
Precisely. Furthermore, even when promising fungal metabolites are found, they are often produced in vanishingly small quantities. We're talking milligram amounts per liter of liquid culture. That's generally insufficient for advanced research, let alone for industrial-scale drug development needed for clinical trials and eventual marketing.
Scaling up production is a major hurdle then.
A huge one. Scaling up from laboratory flasks to industrial bioreactors presents considerable technical hurdles. Traditional liquid bioreactors, which often work well for bacteria, can face issues with the way fungi grow their highly branched mycelia. This can lead to problems like mycelial clumping, increased broth viscosity, which makes stirring and oxygen transfer difficult. All things that hinder efficient large-scale production. And frankly, the sheer cost associated with producing these complex metabolites in sufficient quantities can also be a significant constraint on development. It can be very expensive.
Okay, so making enough of it is challenge number one. What else?
Then there are purification and isolation complexities. The intricate and often very chemically similar structures of many fungal natural products make their isolation, purification, and characterization from the complex fungal broth quite difficult.
Separating the desired molecule from everything else the fungus makes?
Exactly. While traditional chromatographic methods are used for initial isolation, optimizing these processes to maximize the yield of the specific metabolite you want, or even just understanding the diversity of compounds present, has historically received less attention than, say, finding the initial hit. So, getting a pure compound in large enough amounts remains a painstakingly slow and often inefficient process.
Right. And then there's a clinical side.
Yes, finally, we confront toxicity and clinical translation hurdles. Historically, many fungal metabolites and their synthetic analogs that looked promising initially when tested in human clinical trials have shown limited or no meaningful anti-tumor activity, leading to their discontinuation. A major concern, as with any potential drug, is the toxicity profile.
The side effects.
Exactly. Many fungal-derived agents have reported dose-limiting toxicities. That means the side effects become too severe before you reach the dose needed for the drug to be effective.
So you can't give enough of it to work without causing harm.
Correct. These toxicities can include things like neurotoxicity, various forms of non-myelosuppression, meaning it doesn't necessarily wipe out bone marrow like some chemo, but it causes other serious problems. Poor water solubility, which makes formulation difficult, even retinal toxicity in some cases. This results in narrow therapeutic indexes, limiting their clinical applicability.
And the WHO has concerns too.
Yes, the World Health Organization has highlighted a critical lack of effective medicines and diagnostic tools for invasive fungal diseases in general, and they note that several current antifungal treatments themselves cause serious side effects and frequent drug-drug interactions. This just underscores a broader challenge in developing any fungal-derived agent, whether it's antifungal or anti-cancer. It's a complex safety landscape.
It certainly sounds like an incredibly complex puzzle. So many technical and biological pieces that need to fit together perfectly, from the initial fungal fermentation and extraction all the way to how it ultimately interacts safely and effectively within the complex human body. It's a massive interdisciplinary challenge.
It truly is.
Beyond these formidable scientific hurdles, what about ethical considerations? Are there any when you're exploring potential drugs from a known pathogen like A. flavus? Or public perception challenges, especially given its notorious history with the curse and its role as a food toxin?
Those are very important points. Safety, without question, is paramount. Any therapeutic derived from A. flavus must be rigorously tested and proven to be absolutely free of any toxic impurities, especially aflatoxins.
Complete separation is key.
Absolutely. The asparagomycin work specifically addressed this from the outset by isolating a single pure compound and then modifying it synthetically, carefully ensuring that all aflatoxin pathways were avoided. This distinction must be rigorously maintained throughout the entire drug development process. Pharmaceutical development requires meticulous characterization of the specific modes of action, the complete toxicity profiles, and highly robust manufacturing processes.
Including making sure no fungus or toxin gets into the final product.
Exactly, including stringent sterilization and purification steps to prevent any residual fungal DNA or spore contamination in the final drug product. Regulatory agencies like the FDA will treat asparagomycin analogs as completely new chemical entities. They'll require the full battery of preclinical toxicology studies and extensive human clinical trials, just like any other novel drug candidate, regardless of its origin.
And public perception. The curse connection.
Public communication is definitely a significant factor, particularly given the fungus's deep-seated cursed image and its very real association with food toxins. Researchers and pharmaceutical companies involved must clearly, consistently, and transparently explain that no active toxin, like aflatoxin, is present in the medication.
Emphasizing it's a distinct engineered derivative.
Precisely. That it is a carefully engineered derivative, completely distinct and separated from the dangerous natural product. This clear communication is absolutely essential to manage public perception and build crucial trust.
What about bioethics?
From a bioethical standpoint, since A. flavus is a microbe, not a human or even typically an animal subject in the early stages, standard animal welfare and safety regulations apply during testing. But there are no human subject ethical issues beyond the normal protocols for any new drug development. The biggest ethical considerations in this realm might involve things like intellectual property, how do you fairly patent natural products and their derivatives, and potentially considerations of benefit sharing, especially if fungal strains are sourced internationally from biodiversity-rich regions.
Right. Who benefits from discoveries made from resources in certain areas?
Exactly. Interestingly, from a cultural perspective, the idea of the redemption of a pathogen turning a perceived curse into a potential cure has a powerful symbolic appeal. However, this symbolic narrative should never overshadow the objective scientific assessment of risks and benefits. That must remain the primary driver of its development.
So the science has to lead, not the narrative.
Always. It's a journey that certainly demands not just scientific rigor and innovation, but also ethical foresight and very clear communication to build trust, not only in the science itself, but also with the public. The responsibility is squarely on the researchers and developers to be transparent and meticulous.
Definitely.
But despite these formidable challenges, the field of fungal natural product drug discovery is ripe with unprecedented opportunities. The discovery of asparagomycins from Aspergillus flavus is really a resounding testament to the vast, untapped potential that lies hidden within the microbial world.
A proof of concept, almost.
In many ways, yes. And it's not an isolated finding. Researchers have already identified similar clusters of genes encoding RIPPs and other fungi, suggesting that many, many more fungal RIPPs remain to be discovered, patiently waiting for us to find the keys to unlock them.
So asparagomycins might be just the tip of the iceberg for fungal RIPPs.
That's the hope. Kiyu Ni, one of the key researchers on the asparagacin paper, emphasizes that even though only a few fungal RPPs have been found, almost all of them have strong bioactivity. This indicates that this is an unexplored region with tremendous potential, significantly broadening the scope of fungal natural product discovery beyond just this one example.
And beyond IPPs, is A. flavus being explored for other compounds too?
Yes, absolutely. Continued research into other Aspergillus flavus secondary metabolites beyond just asparagomycins also holds immense promise. Studies have already explored other compounds like vitexin, ergonovine, various triterpenoids, luteolin, and phycostrols derived from A. flavus for their therapeutic potential.
Including other anti-cancer possibilities.
Yes, including potential anti-breast cancer activity through targeting crucial tumor suppressor proteins like BRCA1, BRCA2, and TP53, which are vital for DNA repair and cell growth control. So the fungus might hold multiple potential keys.
And technology must be helping accelerate this.
Tremendously. The integration of advanced technologies is key. Things like bioinformatics for sifting through massive biological data sets, gene editing technologies like CRISPR, for instance, to perhaps create fungal strains that cannot produce toxins, making them safer production platforms in artificial intelligence, AI, are all poised to dramatically accelerate the discovery and development of novel fungal RPPs and other natural products.
How can AI help specifically?
AI, for example, can facilitate the swift identification of novel potential drug candidates from genomic or metabolomic data. It might even help predict toxicity or enable the personalization of therapies by analyzing patient-specific data, potentially enhancing treatment outcomes and reducing adverse effects down the line.
The future looks promising then, despite the challenges.
It does. As Sherry Gao aptly stated, fungi gave us penicillin. These results show that many more medicines derived from natural products remain to be found. It's up to us to uncover the secrets. This isn't just a hopeful statement. It's really a profound call to action, recognizing the vast chemical diversity that nature has perfected over millennia, which is still waiting for us to understand and translate into human benefit.
This story truly symbolizes how even centuries-old mysteries, things once shrouded in fear and superstition like the Pharaoh's curse, can unexpectedly lead to cutting-edge 21st century cures. It reveals the incredible wisdom hidden within the natural world, just waiting for us to find it. A journey from ancient legend to modern lab, full of twists, turns, and constant discovery. Hashtag tag tag outro. So what does this incredible deep dive ultimately mean for us? Where does it leave us? We started with the whispers of the Pharaoh's curse, didn't we? A chilling tale of ancient doom.
Rooted in mystery and fear.
Exactly. We then followed the scientific breadcrumbs, moving meticulously from those historical incidents like the opening of Tutankhamun's tomb and the tragic Casimir V's in Poland to the rigorous identification of Aspergillus flavus as a very real, tangible biological threat.
Capable of causing serious illness and widespread food contamination. A genuine danger.
Right. And then we witnessed this profound transformation. how cutting-edge research, really driven by curiosity, persistence, and innovative techniques, turned this feared fungus into a potential source of hope in the fight against cancer.
It's quite the narrative arc. The remarkable journey of aspergillomycins, from being just a unique fungal peptide, initially inactive even, to becoming a potent anti-leukemia agent in the lab.
Yeah.
It truly underscores nature's inherent duality.
How microbes can kill.
But also with ingenuity, deep scientific exploration, and importantly, a willingness to challenge our own assumptions, they can also heal.
This deep dive has truly shown us that even in the most notorious places, like a mold known primarily for causing widespread harm and inspiring ancient curses, there can be a hidden pharmacopoeia, a treasury of therapeutic compounds just waiting to be discovered.
If we have the tools and the vision to look for them.
Exactly. It's a powerful testament to the boundless potential of curiosity and relentless scientific exploration, seamlessly bridging these diverse fields like archaeology, mycology, and pharmacology. Who knew they'd intersect quite like this?
It shows the interconnectedness of scientific inquiry.
It really does. So here's a provocative thought for you, our listeners, to mull over. Consider how many other curses or perceived dangers in the natural world, things once feared or profoundly misunderstood, might actually hold the key to our next great breakthroughs.
That's an interesting question.
What seemingly negative aspect of nature, what overlooked or even reviled organism, be it a fungus, a bacteria, maybe even a toxin, could be the source of a future cure, just waiting for scientific eyes to see its hidden potential and completely transform our understanding?
Where else might cures be hiding in plain sight, disguised as threats?
Exactly. Something to think about. Thank you for joining us on this incredible deep dive today. Stay curious, stay informed, and keep seeking out those surprising facts that truly broaden our understanding of the fascinating and often paradoxical world around us.